Ladies and gentlemen, good evening and welcome. My name is Everall. I'm the Executive Dean of the Institute of Psychiatry, Psychology and Neuroscience here at King's College London, and we are gathering for a great occasion, which is to hear Professor Kei Cho's inaugural lecture. I want to start off by giving you a bit of background about Kei. So Kei was born in Incheon, South Korea, and he tells me in the same year that the Beatles released their Rubber Soul album. And obviously this album is Kei's favourite Beatle albums. What's the track is your favourite?
You will see.
In My Life? He also tells me that his father wanted him to become a diplomat, but clearly that didn't occur. And that Kei actually gravitated towards science, and much of his education was abroad as well. His science and medicine career started at Kyushu University in Japan, and he says to me that he had an artistic way of thinking and was creative and innovative, but this approach wasn't popular during his MSc. So maybe you'll tell us a bit more about that during your talk. He came to the UK to undertake PhD training in 1993, and he finished his PhD at the University of Hull. And explained to me, it's a long story as to how he ended up in East Yorkshire. Maybe he'll tell us more about it later or at the drinks afterwards. But actually he'd grown up there, and he described himself as an innovative and motivated scientist for artistic thinking. He submitted his PhD after just 20 months. And I think that he was completed within 23 months. But you had to wait to have your viva, didn't you? Because one of the external examiners' dog became very ill. And so it was rescheduled 3 months later. His hard work during that PhD resulted in 4 research publications, and he told me that in order to achieve this, he spent most of his time in the lab, including Easter and Christmas. And the great advantage of this, it kept his utility bills down. And so he was able to save money to buy a hi-fi stereo system, which I think is one of your passions. And he's kept this idea all along. So he says he has a similar tactic now, which is don't go on holiday, save money, and buy a high-cost hi-fi stereo system. And you told me that you have approximately 4,000 LPs. Yep. But the aim is to achieve a collection of 10,000. He says, if any of you got any rare LPs, speak to him afterwards, because he'd like to know about potentially collecting them. His first postdoctoral training position was in Durham, and then he later moved to the MRC Centre for Synaptic Plasticity at the University of Bristol. There they concentrate a lot on LTP, long-term potentiation, but obviously being a bit different, Kei didn't do that. He did work on long-term depression and looked at how this kind of caused synaptic weakening. And he found several new LTD induction mechanisms, resulting in 7 further publications, including 2 in Nature Neuroscience over a 4-year period. He then obtained his first academic position, was a lecturer in biomedical science at the University of Sheffield in 2002, and then he was promoted as a professor and chair of neuroscience through the exceptionally talented academic route at the Faculty of Medicine and Dentistry, moving back to Bristol. So he did this bypassing being a senior lecturer or a reader. He was promoted straight to chair. And he tells me he had good research outcomes and did well in the ref evaluations. In 2010, he and his colleagues explored an important biological event that neurons use an apoptotic signal not to trigger cell death, but to reduce synaptic efficiency. And this finding broke the dogma about apoptosis and provided a new theory about synaptic weakening, and this was published in Cell. And this was later translated into aberrant synaptic weakening as a mechanism involved in Alzheimer's disease. In 2011, the importance of his work was recognised by the Royal Society through the Wolfson Research Merit Award, and the great byproduct of this is it kept him in the UK, and so which we're benefiting from today. And then in 2018, he joined the UK DRI hub at King's College London as a professor of neuroscience, and he's currently in the Department of Basic Clinical Neuroscience. He's also the co-founder of the UK Career Neuroscience Symposium together with Professor John O'Keefe, who was awarded the Nobel Prize for Physiology of Medicine in 2014. And this symposium forms a very good bridge between neuroscience in the UK and in Korea. And it is a great link between the two. So this evening, we're going to hear Kei talk in his inaugural lecture about his work. So please join me in welcoming to give the lecture on the journey from synapse physiology to drug discovery for Alzheimer's disease. Kei, welcome. Thank you.
Back to the early 1990s, as you may know, the Kyle and Golgi, these 2 gentlemen scientists, won the Nobel Prize because they found the how the neuron or neurons structured and networked. By the way, the Golgi is an Italian neuropathologist still reusing Golgi staining, which is a remarkable finding. So that was actually the fact. The scientists show how brain or how nervous system will connect and work. That was in 1906, the Nobel Prize. And for a while later, actually, now Hebb, the Donald Hebb, Canadian psychologist, making sort of postulation how brain will work. So-called, yes, Kyle and the Golgi found how brain will be networking or nervous cells will be networking each other. But Donald Hebb actually start thinking how they will work as a brain function. This is so-called neurons or nerve cells fires together and wire together. That is a so-called Hebb's rule. And from up to now, the Hebb's rule is one of the fundamental postulation of the theoretical approach or thinking of the memory. So such as input and output neurons, they communicate through the activity, and the activity will be changed to a certain outcome, like either strong or weak. So this change of the weight or communication is part of the memory processing. So if you look at the more specific or microstructure domain of the neuron, they actually communicate through a certain way we call dendritic spines. So this dendritic spines has to be communicated and change over a certain way. These Hebb's rules, by the way, is still working as a very fundamental finding even now, like machine learning based on these Hebb's rules theory. So that was I was interested in, and so far I try to work on how memory can process and how memory can be preserved and why memory could be disappeared. But to get this journey is very, very humble. So when this is my postgrad, like a master student picture just after a journal club. And I just took the photograph, a little bit depressed, but a little bit tired. And one of my friends that took the photograph at the time. So I tell you why I came to UK, because there's a three gentlemen's fault. Okay, I say three gentlemen's fault. So I presented three journal club papers, and all of them was a troubles. So first the trouble was Timbliss and Teroromos, the classic 1973 synaptic plasticity finding using the rabbit model in hippocampus. So when I saw this paper, I thought, my goodness, this is amazing, because Kyle and Golgi, they found the structure, and Hebb's rule actually postulated a certain way, but this is the first time actually showing this through the experiment, showing that will be the true. So I was very excited. That's why I chose this journal and then presented it. So the journal is very simple. They're using this, the prep, and then deliver a certain activity in input side, and then measure postsynaptic as the output side of the synaptic weight, whether they change or not change. So-called neurons fire together and wire together. That is Hebb's rule. So Tim and Thermo, they deliver certain this electric stimulation, and then you can see that after stimulation, the synapse, the amplitude change. So such as this become like big, and this is unconditioned, unstimulated pathway input, they didn't change. Clearly, activity or experience will change wire, the synaptic transmissions or synaptic weight. That is exactly what the Hebb's postulated early days. And my supervisor made a criticism, Kei, look, they're using bloody high frequencies. That cause artifact. So this one and that one is probably not comparable because of there's so many factors to change it. So I was a bit depressed because I was so excited, but the comment was, well, fair enough, at a certain point, yes. So next round, I present another paper, which is 1983, Graham Collingridge's using one of the glutamate receptor antagonists, AP5, which is an NMD receptor antagonist. Actually, that antagonist block the potentiation and then wash out it and then deliver another high frequency. Now they have a potentiation, which is a bigger synaptic weight. So which suggests clearly it's not just artifact. There's something real things happen through the activity. So I presented this, and my supervisor wasn't that happy because it looks like I'm just against him. But he didn't give any comment. So my third round, I presented this, which is Richard Morris 1986 and published in Nature, using exactly the same AP5 infused and measuring the famous Morris water maze, but also performed electrophysiology whether they can see the LTP kind of synaptic change. The AP5 block the LTP induction, but same time, AP5 actually making worse in terms of the learning and memory. As you can see, this is control animal, the drop, and then the easy to find the target podium. This is the AP5 animal never actually found where the podium was. So clearly, yes, plasticity and memory somehow is correlated. So at this point, actually my supervisor completely crossed. And he said, comment, Kei, I give you a kind of advice. Why don't you go to UK and do the more research? That's why I came here. By the way, that three papers, or three gentlemen won the Brain Prize year 2016. So I think that is a remarkable finding. And from my young age as a scientist, I completely inspired these three gentlemen. And still, I have a good relationship with this gentleman. Every single time, whenever I saw these three gentlemen, that is your fault. Okay. So I joined kind of a dream, someone who actually passionate about synaptic plasticity, tried to join where the synaptic plasticity is found. So year 1998, I had a chance to join the MRC Center for Synaptic Plasticity. So I thought I could now work together with this LTP but I couldn't actually working on the LTP, but I did a lot of LTD work, long-term depression. So what I said, my standing position is both LTP and LTD is important. Of course, LTP and LTD is a slightly different or quite different, but they share quite similar concept. But briefly, when the LTP and LTD determine so-called the fate, determine a direction of synaptic plasticity, one is getting bigger only, or a certain way is making more available glutamate receptor in the surface. That means they change the conductance more like a bigger and large conductance. Opposite way is LTD, is reduce the number of available receptor or reduce conductance, and then they can be like a weak. So one is probably positive side, one is probably negative side, but it's not necessarily. For example, like pain research, for example, maybe LTD is very positive side of plasticity rather than negative side. So it is like LTP and LTD. It looks like very different outside, but if you look at inside, very similar, such as like it is involved glutamate receptor, like NMD receptor, and it is involved calcium mobilization, and certain enzymes, they share exactly the same. It's very, very hard. If you hit LTP or if you hit LTD in PubMed, you will end up with nearly 70% enzyme or signaling. They just share exactly the same. So I was very curious, and then how many systems or how many signals or how many mechanisms actually involved triggering this LTD kind of synaptic plasticity. But I just make the example of what I found so far, and it looks like just endless. Certain point, I met Professor Morgan Sheng, and we met in the Society for Neuroscience meeting in the United States. And we sat together and say, Kei, I know you're doing a lot of LTD. What he said was LTD equals cell death. So I'm arguing, well, no, LTD is kind of cell death, but it's not necessarily cell death. It's part of the synaptic plasticity. That is like a sort of physiological consequence rather than pathology. So we debated. Long journey in the sitting in the New Orleans airport, and the decision was, why don't we actually try to figure out, is this cell death or part of synaptic plasticity? So that actually changed my scientific vision, and we published the new conceptual understanding. Apoptosis is not necessarily kill the neuron or kill the cell, but certain way it actually minimizes synaptic plasticity or certain signaling pathways. So he was right and I was right. Both was right. This paper now building up to the next level. Wow, we thought apoptosis is actually equals bad. Apoptosis equals cell death. But how they can manage, or does it mean physiology and pathophysiology can coexist together? Or one to the other, one of the balance is the breakdown, and then probably it goes to wrong direction, such as like pathology. So just long story short, one of the fundamental findings was the caspase is involved in cell death, but not necessarily, but caspase we found is involved synapse weakening, like LTD kind of change. So this is a caspase 3 knockout mice. We cannot see LTD kind of plasticity, but LTP, so-called positive plasticity, is actually normal. So that means I show you the LTP and LTD twins. It looks like exactly different, but they share the certain signaling. We found, oh, that maybe now we crack down certain code. What is distinct between LTP and LTD? So when the presynaptic terminal released the glutamate and NMD receptor activated it, we believe this NMD receptor actually stimulated mitochondria and then released the cytochrome C and activate caspase 3. That caspase 3 somehow causes AMPA receptor endocytosis or weakening. Later, we puzzle all this together and then find out there's certain balance between weakening versus strength. So-called positive versus negative, they're balancing all the time, but if they break the balance, it goes to one direction. So activity or activator somehow activate caspase signaling cascade. And caspase, by the way, this guy is continuously cleaved AKT1, which is a survival signaling cascade. On the other hand, this AKT1 chronically or permanently suppress GSK3. So somehow positive signal versus negative signal, if they're balancing, or making the break point, but if one break point is disappeared, and then it goes to the turning to the other way, such as if strength pathway is strong and then weakening pathway is suppressed, or vice versa. One important thing is this GSK3 is also found the key LTD element by Professor Graham Collins' lab. So which means somehow caspase and GSK3, they are in the same line of synapse weakening pathway. So we postulated, wow, that means somehow this caspase, AKT1, GSK3, the triangles, these guys has to be worked together, and somehow breakpoint, it goes to one direction to the other. Interestingly, this GSK3 is typical tau kinases, so that causes tau phosphorylation. So from the basal, it depends on how the signal balancing between strength or weakening, and then it goes to one to the other directions. So we published a cell paper with several couple of years of the pain, but we end up with a very good outcome. And another point is, so what? So what? We have another cell paper, another Nature Neuroscience, another neuron paper. So what? We want to do another mechanism, another metabotropic or muscular receptor. There's a bunch of mechanisms come to the other. So I and Morgan sat together and, do you want another paper? No. So why don't we think about what we can do with what we know about the synapse weakening or LTD mechanism? So-called science never reached the therapy if we do a right bridge. Otherwise, science is one direction, therapy or therapeutic demand is the other direction. So-called we need to translate. So this is a Bristol suspension bridge, by the way. So it has to be link those two values. Particularly, I was interested in, well, including everybody, the memory and why we're losing the memory. It's not just simply age issues, but also there's certain disease causes memory deficit. I try to emphasize, I'm not gene targeting or gene therapy kind of things, but just fact. Just forget about all numbers. I would like to highlight one point here. This is when I was the sabbatical in Cambridge many years ago. This is one of the college, the master, he was a mathematician. And over the lunch every single time, I had to learn new language, which is mathematicians never understand proteins, vice versa. And we actually found something very interesting, common sense. How do we know certain disease, how much that will cost to cure or care? But we can calculate in Britain, but that goes to South Korea or Japan or United States or New Zealand. The figure is completely different. So 1 month I'm working on with these mathematicians and so-called dependent index. I made the equation. So for example, you got a cold. What is a dependent index? 0. Which means you don't need any help. Just rest yourself, couple of days, drink a lot of water, vitamin or fruits. Several days, you don't need any medicine or special care. 0. Cancer, 0.26. So we are living 24 hours society, so we can't work 24 hours, which means in hospital or medical care, there's a 3 shift. So 0.3 means 1 person, okay? That is the dependent index curve. Dementia or Alzheimer's disease is 0.85, which is nearly 3 times of cancer. That means at least 1 patient, 2 or 3 medical staff should be there. That is dementia. So I was so interested in, based on, not just based on this, but also as overall, because of the memory was my top priority in terms of the research. So looking at this LTD kind of concept, how we translate this to the disease and therapy. As you know, the two landmark feature of the Alzheimer's disease, by the way, Alzheimer's disease causes dementia. Most of Alzheimer's disease, the dementia is caused by Alzheimer's disease. And there's a two landmark feature, which is amyloid plaque, which is protein misfolded outside of the cell, neuron, and also neurofibril tangles, which is hyperphosphorylated tau that building up inside of a neuron, and both cause a toxicity. So year 2011, I gave my inaugural lecture in Bristol. This figure at the time I searched. Now this is yesterday I searched. So the A-beta related publication run up 233% and tau is so on. And others surprisingly now catch up, which is very rapidly building up as a key or possible target for the Alzheimer's disease or cure. So if you want to be a PI or independent, there's a scope, others is more chance. Now, problem is if someone asking very cynically, what is the roadmap for the cure, clinical applications? As you can see, there's no real answer. Okay. Point is because of, it doesn't matter how many papers we published because of the loss in translation. That is a fact. So I look at all literature and making some assumption, at least a tau and amyloid beta context. So arguably, the amyloid plaques, no doubt, but amyloid plaque building up early stage, and then fibril tau, fibril tangle coming building up later. Interestingly, somehow symptom is definitely correlated neuronal integrity. So this suggests that somehow what we should really think about is functionality change and how that functional change actually impact learning and memory, such as like cognition in this case. So we postulated definitely integrity is caused more likely the symptom degree, the change of the symptomatic degree. Therefore, we think the synapse weakening or functional weakening probably part of or main feature of the pathophysiology. So many scientists, last 10 years, Alzheimer's disease, synapse elimination, cell death, it is true. And then they look at the genes, it is true. But what we didn't actually look at is what actually happened very early stage and how they change. Dead neuron, what you want to do? There's a dead. There's no way to reverse it. But if symptomatic change or functional change, there probably is small hope or therapeutic window is available, or slow down going to this end point. So that was our rationale to working on the synapse weakening pathways, how that can be translated as a therapeutic target. So first approach is immediately after our the caspase cell paper, we're simultaneously working on together with this amyloid beta model. As I said, caspase is one of the bad guy. If caspase is activated, and then GSK, the GSK can become a more proactive that cause more negative side of plasticity and actually inhibit positive side of plasticity, such as blocking LTP kind of plasticity, but enhanced LTD, also called synapse weakening. The question mark is, can we stop these guys here or blocking GSK, or somehow caspase cannot cleave AKT1, then can we prevent or reverse it? This paper suggested yes, indeed. So somehow synapse weakening pathway, we could reverse some functional functional problems, such as we can protect LTP from pathogens, in this case, amyloid beta. So it doesn't matter amyloid or other pathogens. As brought again this diagram, weakening versus strength, balance is coming from which signal actually triggered by certain pathogens. And can we reverse it, such as like from basal more stabilized or aberrant synaptic weakening, which is losing certain functions. And key of this is the tau and tau phosphorylation. So I had another very artistic way of thinking, and even more than me, one of the young gentlemen actually rang me and emailed me and suggest a certain summer internship. At the time, I have no idea what he's talking about. And it's all about this, the machine learning and the simulation. He's a computer scientist. So now, even now, still I could understand only I request a certain slide and he gives some idea how to prepare today's slide. So I will do my best. So data mining, as I said, the iceberg, top of the iceberg, more than today, yesterday's the PubMed count is more than 100,000 publications, well over 100,000. So some information is available already. So we don't need to repeat certain experiment again and again and again using this data mining. So once we accumulate all these or parameters or certain concept what you are interested in, now computer will sort out your problems. And then it brings to certain key word. Now there's a ranking 1 to ranking 100, what you want. Okay, and then you choose, oh, I don't like ALS, so I delete ALS. Or I don't like amyloid beta, then I can delete. So that is like a so-called sorting, and then after sorting, and they come up with some data evaluation and then endpoint chance. So this gentleman's first question was, okay, you are interested in caspase AKT1 GSK3. What is the chance that actually happen synapse weakening? My goodness. So he wants numeric number rather than is the significance. Because computer scientists 0, 1, 0, 1, they like numbers. Just simple as that. I couldn't answer what is a chance numeric number. So he sorted out the real number. So this is a model what I'm interested in, and then sorting what is available, already known, protein-protein interactions, and so on. And pick 3 very interesting molecules. One is caspase-3, GSK3, and PS95. There's some more, but this is so-called biased sorting. And then making prediction like endpoint parameters. Endpoint parameters, synapse weakening, synapse elimination through synapse weakening. So what we found was if synapse weakening pathways, the chance, more than 80% chance in Alzheimer's disease actually happen. And once the synapse weakening happened, and the consequent synapse elimination is more than 60% chance, definitely synapse elimination is going on. Now, very striking finding was if stop this more than 80% chance, then synapse elimination is reduced like 80%. So that is remarkable. And then he drew me a very interesting model. So who likes football game? Not rugby, football. Okay.
to win the football, or never lose a football game, what is the best way to do it? Never lose. Well, it's very similar. If you don't have goalpost, you never lose. Alternatively, if you have a great goalkeeper, so his success rate is 100%, you never lose. So he brought the list and then identified two molecules. One is a tau, and one is something else. I can't tell you because the video is like a take it. So tau is top listed molecule, like either you don't have any goalpost, or if you maneuver something, that goalkeeper, keep all balls. So we studied tau, and what we found was there's a two dogmatic the theory. One, tau is a microtubule protein, therefore it's more likely axonal protein, but the reality is not. This is a gold labeling EM study showing, this is by the way from mouse. The wild type mouse, you can see the presynaptic terminal and postsynaptic region, you see both gold labeled tau. This is tau knockout mice. Obviously you can't see any tau. So tau is everywhere, particularly synapse. And what we found in this paper, tau is associated physiological, but also the possibly pathophysiological synapse weakening. And later, also we found this tau is associated or interact with AMPA receptor endocytosis, which is they can reduce, tone down certain activity through these molecular pathways. So somehow the wild type mouse, when we deliver NMDA, in general, this NMDA causes AMPA receptor endocytosis, like from surface into the cytosol. But tau knockout, we can't see any certain molecular interactions. That means somehow tau has to be there. Otherwise, if tau is not exist, then AMPA receptor endocytosis won't happen. So we try to go even a little bit more further using several different tools. By the way, this gene-gun approach is like a Lego puzzle. We can customize what you want to knock down, but at the same time overexpress the same cells. So it's a great tool, and lucky enough, I have a great postdoc student. They work on very well, so we have a great chance of success. Now, using this tool, what we've done was we're knocking down endogenous tau, for example, and overexpress human forms of tau, but several different mutated forms, such as like serine phosphorylation site. So we identify one single phosphor residue, which is serine 396, is important for the synapse weakening by tau. Now, if that is the case, then how tau can be interact with the weakening? Of course, the synapse weakening, or sorry, the AMPA receptor endocytosis or trafficking, there's a lot of complex of the multiple stage of processing to deliver it. One idea is somehow tau link or interact with key endocytotic molecule, which is we call the PIC1. If PIC1 somehow interact with tau, then maybe it will happen that way of the synapse weakening. Interestingly, this protein X, not just the functional, but also the structure of dendritic spines, which is a fundamental for the synaptic transmissions. So we can identify like a stubby, like a big or thin or mushroom-shaped spine. This, by the way, this is live CA1 neuron from hippocampus. So we can actually see and count whether they change the shape. For example, many people agree the mushroom shape is a very positive side of dendritic spine, and thin is like either weakening or sort of negative side of the shape. So we can count sorting how many types or what kind of population we have through the different conditions. So long story short, when tau phosphorylated, they're losing the mushroom shape and a more thin shape, which is positive side of the dendritic spine changed to negative side of the dendritic spine. Now, interestingly, when we knocking down protein X, which is cause the synapse weakening through the AMPA receptor endocytosis, then that phenomena actually reverse it. You can see this is a thin shape now, sort of the reduce reverse it, and this is a mushroom shape now back to control levels. So we believe this is a very good first step to identify a certain target, not other enzymes or A beta mediated, but this is a tau specific target for Alzheimer's disease or dementia to prevent the synapse weakening phenomenon. Last year I joined DRI. Thanks very much, Chris recruited me. And this is based on synapse weakening theory and postulation. We try to understand whether we can identify certain therapeutic window, that therapeutic window can be actually work for drug target. So I emphasize already, if already permanently synapse elimination happened, there's no way to reverse. The chances, I think, is somewhere in between here. Now, this bar indicate what possibly happened in the biology. Physiology happened from the beginning, but when physiology is losing their physiology, actually building up the pathology or pathophysiology, maybe physiology and pathophysiology coexist from the beginning, but somehow it tunes which direction they actually goes. So this is my aim and objectives. We try to building up more understanding very early stage and then making key target of drug discovery. Well, it's not just a neuron. The brain has environment. So we also pay attention what other factors or factor could be regulate these weakening pathways, including like microglia cells and others. So we are thinking about what else we can actually do. For example, our life patterns, maybe change this, our brain environment, and that could be another way we have to think about it. Now, there's a final several slides left. I think I took too much time, but the Royal Society and myself is something very special. There's several reasons. And when I had some very interesting idea when I was in postdoc, I didn't realize postdoc has a so long annual leave because I never use annual leave. And I have a very interesting idea, but I don't want to stop what existing project. So I went to the, at the time my mentor was Malcolm Brown, he retired. Malcolm, I really have this idea, but I don't want to disturb your project. And he smiled at me and, Kei, you never use annual leave. And, oh, do I have annual leave? How long? And he said, you have more than 3 weeks. Oh my God, I have a 3 weeks annual leave, more than. So I took annual leave and I performed a certain experiment, but I don't have my grant because at the time I was postdoc. So I found the Royal Society have a travel grant, research travel grant. I'm sure the John, you're my reviewer, I don't know. But I'm lucky enough, every single time I got this fund and I made one remarkable finding, but also carry on my research. The other occasion was when I become a lecturer. One of the difficulty is junior lecturer, very hard to get research grant. Wellcome Trust rejected, and MRC rejected. Royal Society, well, not big money, but we can do something. And actually, we published Nature Neuroscience. It's not bad, okay? So I will tell you, one of my artistic way of thinking is this. We are living 24 hours, day and night. But you think about if you are wrong place at wrong time, like I'm sure Chris has a lot of jet lag, or not. If we have this wrong time, wrong place, because our body has a clock, but our external environment has a clock, but it's these two clocks when your same environment is always synchronized. But if you go to Japan or LA and then start, external clock and your body clock is completely dissociated. And then it causes stress. So when I took the airplane, cabin crew didn't bring my orange juice. I asked 3 times. So I was completely crossed. Do you have any memory problem? Oh, how do you know that? So I have an interview, and then I found, well, they have some problem, their cognition or memory. This data suggests actually they have a certain either temporal or permanent deficit. I don't know yet, but they have definitely, they have some cognitive deficit. This paper, because of the Royal Society travel grant, this can be happen. And you can see only one author, which is me. So postdocs, please using your annual leave for your research. And that paper came out year 2001. This is just a day before general election in UK. Times newspaper, front page. I can show you what is the front page says. This is what Times newspaper in the front page said just day before the general election. Remarkable. Furthermore, Washington Post. This is longer than my original article. I don't know how they make this, big article. Okay, there was a brief communication, only 1.5 pages. But anyway, this columnist making a very nice story. I think that's why I'm John. Your FRS, thanks the Royal Society all the time. This is my heart all the time. And of course, my team and my collaborators, all the time they give a lot of support, and of course, the funders. But importantly, Chris, you recruit me, and that's why I'm here. And I'm really sincerely thanks all of you, particularly Ian and Chris. Thank you so much. Thank you.